Okay, so welcome. So it's always a distinct pleasure to have our annual George Corey lecture. And some of you, the older colleagues in the room, remember George. Some of you younger colleagues probably just think that this is a great lecture and it's a wonderful opportunity to come and have some free coffee and cookies. Incidentally, there will be some in the reception afterwards. So, but indeed, on the screen you see a picture of George. This was taken in 1987. And George, at that time, was remarkably young, was only age 43, and was elected at that time to the National Academy of Science. So indeed, in a very short period of time, had really accomplished a tremendous amount, both in terms of science and some of us remember also in terms of his mentorship. So several of us in the audience, including myself, was trained by George, and we were, of course, very grateful for the training and his mentoring, and he was, in fact, a truly, truly remarkable scientist and a teacher. And some of you may recall that nowadays when you work with sort of DNA binding proteins, transcription factors, and enhancers, you think that they were just there. But in fact, that entire concept of enhancers and transcriptional activators was in fact originally discovered by George Corey. And in fact, in honor of George's memory, we actually started this lecture, and it was started now as far back as 1994. And during all the different episodes of government shutdowns and whatnots, the lecture has managed to persevere. So we've had a number of stars, including the very first lecturer was Arnie Levine, and in the intervening time, as you can see, some of the other cast of outstanding scientists who've come and delivered the lecture, including Bob Weinberg, Peter Howley, Bob Tijan, David Baltimore, Phil Sharp, Bert Vogelstein, and the list goes on and on. So here I just show you some of the examples of really outstanding speakers who've come to give the Corey lecture. And today, of course, is no exception. And so if I could, then what I would like to do is to introduce our speaker today, and he is Jeremy Nathans from Johns Hopkins. And Jeremy, of course, is also one of these young and wonderful, outstanding scientists. I think he was also elected to the National Academy, probably not much older than 43, right? Perhaps even earlier than that. And Jeremy is, of course, known for many of you for his work on the research in the molecular biology of vision. And he was the first person to isolate the genes that code for human visual pigments, discovered the first sequences of eukaryotic sensory receptors. He taught us the molecular basis of inherited variation in color vision and defined the biochemical basis of different forms of macular degeneration. So vision is something that's very important to us, and I think this is something that all of us, it behooves all of us to benefit from Jeremy's work and from what he's here to tell us. My fond recollection of Jeremy was seeing him, the first week when I was in college at MIT, seeing this tall, smart, lanky, student who was in my recitation class at MIT. And, 30 years later, he remains tall, lanky, and even more smart. So, welcome, Jeremy.
Well, T, thank you for that very kind introduction. I think I have my own mic at this point, so I can speak like this. It's a great honor to be here, especially to give the George Corey lecture. I never met George Corey, but I feel I have a certain connection with him. He collaborated with my father, Dan Nathans, in the 1970s on SV40. And with Malcolm Martin, they mapped the first transcription maps of that viral genome. And there are a series of papers from the mid-70s on which they are co-authors, which really, I think, laid out in many respects this paradigm of dissection of genes and chromosomes. And Dan always spoke of George with great affection and respect, and so I sense a certain connection even over these many years. So let me describe for you some of the work that we've been doing now on the evolution of primate trichromacy. And I want to start with two somewhat extended appetizers before we get to the main course. The first appetizer will be historical. I'm going to look at the history of color vision research very briefly. The second will be to look at variation in human color vision. And then we'll get to the main course, the evolution in primates. So one might ask, actually before we do that, I'm going to just, before we get to the meat here, I'm going to just mention some names so when I describe their work during the lecture, it will connect. And in particular, I want to mention people in my lab who've worked over the years on color vision: Hugh Cahill, Jenny Mackey, Shanath Merbs, Phil Smallwood, Hui Sun, Nianshu Wang, and Chuck Weitz. And we've had many wonderful collaborators, in particular Jerry Jacobs. So I like to do that at the beginning so the names are up there. Now let's just ask, why study color vision? The obvious answer is because it's beautiful. And the world, as we see it up here, looks a lot more beautiful than if we didn't have color vision. And it would look like this if we didn't. But of course, there is a Darwinian reason that nature has color vision, and that is that we can discriminate objects better. So we can discriminate based on chromatic composition of the light coming at us, that is, the reflectances of the objects, better if we have this discriminatory power than if we do not. Now the story, the first of our appetizers, the historical one, begins, in 1665 with the work of Isaac Newton. Newton was at that time an undergraduate at Cambridge University, and he did a very simple experiment. He drilled a small hole in the wooden shutter of his dormitory window to allow a beam of sunlight, and he passed that beam of sunlight through a prism. So for those of us of a certain age, this looks like the Pink Floyd album, Dark Side of the Moon. Perhaps I'm dating myself. And so this beam of sunlight was refracted into its component colors: red, orange, yellow, green, blue, violet, with red being the least refrangible, that is bent least, and violet bent most. And Newton realized that this wasn't just a pretty party trick, it had been for millennia, but this could be used to purify the different types of light, that is different by virtue of their angle of refraction. And that is, if he built a screen with a small hole in it, he could allow through that small hole just one particular kind of light to pass through. Of course, today we would do this with a tunable laser. And then Newton further appreciated that once he had those different beams of light, so he would do this with multiple positions in the spectrum, he could do various interesting things with them. So let me just show you, before I show you one of his experiments, show you his little pencil sketch. This is Newton's own sketch of the setup. This is the hole in the window. Here's the sunlight. It's hitting the prism and the different points of light that are striking, the rainbow colors that are striking this backstop here are mostly stopped, but here's one of them that's allowed to go through, and it goes through another prism over here. I'll just mention parenthetically that among the many things that Newton discovered, remember he was an undergrad at this time, was he noticed that if he put a piece of black paper just beyond the red light, where there was no apparent light shining at all, the paper warmed up. And he correctly surmised that there were lights, infrared that is, that we cannot see, but that physically exist. So that was the discovery of infrared radiation. But for the purposes of this talk, the interesting experiment is this one. And here I'm showing it as we would do it today with projectors. If you project a spectrally pure red light and a spectrally pure green light and superimpose them, you will see something which gives the sensation different from either. That is, it looks yellow. Now, Newton did this experiment. This yellow is indistinguishable from the yellow that you would get by a pure slice of the spectrum at a point intermediate between the red and green points. And the question is this yellow identical physically to the spectrally pure yellow? And the answer Newton showed was no, it is not, although it's identical in appearance. How can you show that it isn't? Well, you can pass it again through another prism. And when you do, this will split up into its component red and green lights, whereas a spectrally pure yellow remains homogeneous with the same angle of refraction. So again, a very fundamental principle elucidated, that is, the human eye is incapable of discriminating certain physically different stimuli in the color domain. And we'll, in a second, get to why that is so. Now, Newton had a number of things to say about this phenomenon, and let me just show you one of them. This is from his Optics, written later in life. And here he's comparing color vision and the sensation of sound, the auditory sense. And here's what he says. May not the harmony and discord, that is the similarities and differences of colors, arise from the proportions of the vibrations propagated through the fibers of the optic nerves into the brain, as the harmony and discord of sounds arise from the proportions of the vibrations of the air? Now, this is an amazing insight. At a time, a century before, it was clear that light had wave properties, before the famous Young interference experiment. So Newton is reasoning by analogy that light may have, in different wavelengths of different colors of light, may have different wavelengths and have some sort of intrinsic wave attribute. Let me also just note that this particular point in Newton's life, the 1660s, was an especially fertile one. In fact, 1665 was arguably one of the most fertile years that any scientist has ever had. And I'll just read you a little reminiscence that Newton wrote later in life. He says, in November of 1665, I had the direct method of fluxions. That's what we call today differential calculus. And the next year in January, had the theory of colors. And in May, I had entrance to the inverse method of fluxions. That's what we call integral calculus. And in the same year, I began thinking of gravity extending to the moon, and from Kepler's rule, I deduced that the forces which keep the planets in their orbits must be reciprocally as the squares of their distances from the centers about which they revolve. That was a very good year. Now, despite his enormous physical insights, Newton actually never spent much time, as far as we know, thinking about or investigating the physiologic basis for these color mixing experiments that he did. The real insight came from Thomas Young, a century and a half later, who was a great physicist, physician, linguist. He did many things in his life. And in his 20s, when he was delivering the Bakerian lecture to the Royal Society, mostly about things unrelated to color, he had one particularly interesting sentence thrown into this lecture, and it's as follows. Here he's talking about the receptors for color vision to explain this phenomenon of color mixing. He says, as it is almost impossible to conceive each sensitive point of the retina to contain an infinite number of particles, by which he means receptors, each capable of vibrating in perfect unison with every possible undulation. So at this point, Young had done the famous interferometry experiment. He knew that light had wave properties, and so he's thinking of these undulations, these waves, are the things that are going to stimulate the receptors. He says it becomes necessary to suppose the number limited, for instance, to the three principal colors, red, yellow, and blue, and that each particle is capable of being put in motion more or less forcibly by undulations differing less or more from perfect unison. That is, he's suggesting that each receptor is a broadly tuned, bell-shaped curve with respect to wavelength, and depending where the stimulating light resides on that wavelength axis, it will stimulate the receptor more or less. And that is exactly right. So that idea was confirmed about 50 years later by the work of von Helmholtz, shown on the left. He's in his mid-20s here. He just invented the ophthalmoscope at the time of this photograph. And here's James Clerk Maxwell on the right, also in his 20s. He's holding his famous color top, this color wheel, which he used to mix colors. He'd been fascinated by color vision since he was a youngster. And the two of them together put the Young theory on solid footing. Now, if we flash forward by 50 years, where the histologists have now entered the picture, it became clear that the retina was a thin layer of neural tissue on the back of the eye and had distinct photoreceptor molecules. This is from 1887, still quite accurate. And there are both rods and cones, and the cones are the ones that mediate color vision. And if we flash forward another 50 years, Ruth Hubbard and George Wald showed that the light-sensing molecule is a protein, so-called opsin, which is covalently bound to a chromophore 11-cis retinal, shown here. It's a covalent attachment to the amino group of a lysine. And the effect of light is to isomerize this double bond from cis to trans. That's the stimulating event in vision. And now, fast forward another 30 years. At this point, molecular genetics arrived on the scene, and I was fortunate to be in the right place at the right time at Stanford University in the 80s, the early 80s, and picked up this problem and identified and sequenced the genes for the human visual pigments. And we have 4 light-sensing proteins. Each binds 11-cis retinal, and each has the same overall 7 transmembrane topography. They're G-protein coupled receptors. And in this pairwise comparison, we see the short wavelength sensor, so-called S, versus rhodopsin, the one that mediates dim light vision, where every filled ball indicates an amino acid difference. And here is the medium wave sensor in the medium wave cones versus rhodopsin. Here's the medium wave sensor versus the short wave sensor. So we have 3 cone sensors, 1 rod sensor. And then here, the medium versus the long wave sensor. So in just looking at the sequences of the 4 receptor proteins, there's an immediate asymmetry here, which I think you can appreciate, and that is that the medium and long wave sensors are much more similar to each other than are any of the other comparisons, any of the other pairs. And that turns out to be reflected in the absorbance spectra of the encoded proteins. So this is work that Shauneth Merbs did when she was a student in the lab. She expressed the proteins from their cloned cDNAs, reconstituted each with the chromophore, and took the absorbance spectrum in a spectrophotometer. And here is the S, or shortwave sensor, peaking at around 440 nanometers, rhodopsin at about 500, the M, or medium wave sensor, at about 530, the long wave sensor, L pigment, at about 560 nanometers. And you can appreciate that the M and L pigments are quite similar in their absorbance spectra. They all have the same bell-shaped curve, but they're shifted back and forth along the wavelength axis, depending which protein they bind. So just to connect to that observation of Newton, that certain combinations of lights were indistinguishable, let me just give you Maxwell's explanation for that. Maxwell conceptualized the system in a very simple 3-dimensional format. He suggested that we can just think of the medium wave sensor amplitude of excitation as some value on one axis, the L sensor as another value on a second axis, and the S sensor's value on a third axis. And so the color world is basically a 3-dimensional space, which is defined by the degree of excitation of each sensory receptor type, and every stimulus is some point in that 3-dimensional space. So basically, any particular combination of lights which leads to the same degree of excitation, i.e. maps to the same point in that space, will obviously be identical to our percept. It will yield an identical percept. It won't necessarily be physically identical. It will just map to the same point. Okay, that's appetizer number 1. Let's move on to appetizer number 2, which is inherited variation in human color vision. And this will bear directly on the evolution of color vision in primates. One of the surprises that we got when we identified the DNA segments that encode the human L and M and S pigments is that the L and M pigments are right next to each other. The genes that encode them are right next to each other on the DNA. Here they are in schematic form. 5' is the base of the arrow, 3' is the tip of the arrow. The L pigment is upstream, the M pigment is downstream. And we believe, and I'll justify this in just a few minutes, that they arose by a duplication event in which there was already pre-existing in this species, this ancestor of ours, an L pigment gene and an M pigment gene, and then an erroneous meiotic recombination led to the rearrangement which gave L upstream of M. Now, the genes are still incredibly similar in sequence. This event must have happened not so very long ago. They are about 98% identical at the DNA level. The region that's duplicated is 39,000 bases. And in fact, because of that duplication, there are continuing rearrangements, meiotic recombination events in which duplicated units are offset. And so one gets not just the basic arrangement of one L and one M, but one L and two M's, one L and three M's, and even 4 or 5. I was the wild type for these studies. I turned out to have one L and two M pigment genes. That's actually the most common arrangement. About 60% of human X chromosomes look like this, about 20% look like this, and the other 20% have additional M pigment genes, one of the most common polymorphisms in our species. And it's just this kind of variation, this unequal recombination, homologous recombination, but offset, that is the source of the genetic mischief that creates the common anomalies of color vision in humans. And the most famous person who had one of these is shown here, John Dalton. Dalton, of course, you know from the unit named after him. The Dalton is the unit of mass in chemistry and physics. Was a remarkable intellectual, lived in the late 18th century in England, and in his first paper, written in his late 20s, actually about himself, which he entitled Extraordinary Facts Relating to the Vision of Colors. I've tried working in words like this into papers that I've submitted to journals like Cell, like extraordinary this or that. They haven't gone very far. And here's what Dalton had to say about his own vision. He says, "That part of the image, which others call red, appears to me little more than a shade or defect of light. After that, the orange, yellow, and green seem one color, making what I should call different shades of yellow." He also noted that he knew of a number of males who had this same problem, but he knew of no females who had it, the first inkling for X-linked inheritance. And what the world looks like to someone like Dalton is shown here. Actually, we now know that Dalton is a so-called deuteranope. He's missing the M pigment gene. But both protanopes, that's the ones missing the L or ones missing the M, have roughly the same defect with respect to discrimination. That is, all of these lovely colors of fruits, orange, yellow, green, and so on, look like a washed out yellow. They all look the same. These are at the long wave end of the spectrum, and you need to have both L and M sensors to discriminate in that region of the spectrum. Okay, so what's going on in those people? Well, what's going on, as I alluded to earlier, and I'll show you now, is that this sort of unequal recombination, which leads to the acquisition of an additional gene by one meiotic product, that's the one shown at the bottom, leads for the other meiotic product, the one that goes like this, to the loss of a gene. So this one has only an L pigment gene remaining. And this can happen both between the genes in the intergenic DNA, or within the genes in the intragenic DNA. The introns are quite large, but again, 98% identical in sequence, and so you can get a recombination, say, in one of the M pigment introns matching up with its homologue in the L pigment, and you'll get a hybrid gene on one chromosome, which has now lost the other genes, and on the other chromosome, the other product, the meiotic product, we get the reciprocal hybrid plus the normal M pigment gene. Just a little detail shown by Samir Deeb in Seattle. It turns out that only the first two genes in the array are expressed. All the distal ones are silent. And I'll show you the reason for that in a second. So this person would make a normal L sensor, but then they would make a hybrid sensor, which, as it turns out, is neither L nor M, but it's something in between. And let me show you the evidence for that in between. Really two pieces of evidence. First, we've produced those pigments. Shaunith Merbs, when she was a student in lab, produced the various hybrids and showed directly in the spectrophotometer that their absorbance spectra are different. In fact, they're intermediate between the two parental pigments. But also, we've gone back and looked in the living human eye to determine their spectra. And we did this by identifying with Ted Sharp and Andrew Stockman a series of men who have just one visual pigment gene on their X chromosome, and it's of a hybrid sort. So we're simplifying the situation as much as possible, looking at males, because then we only have to look at one X chromosome, and looking at males whose X chromosome has just one gene. So this is as simple as it gets. And of course, we sequence the gene, and then we psychophysically test the subjects. And I'll just show you one subset of that data, which is this is a flicker photometry experiment where we're asking the individual to look at a screen on which we're projecting in alternating fashion, 10 or 20 times per second, a spectrally pure light of a known wavelength, say 500 nanometers, alternating with another light of a variable wavelength, say 600, 610, 620, 630, and so on. And we ask the person to adjust the ratio of those two stimulating lights to make the sense of flicker completely disappear. And remember, these individuals have just a single visual pigment in this region of the spectrum. And so for an individual, for example, who has just an M pigment, the curve of sensitivity is shown here, determined by those dial settings which at each of these variable wavelengths have eliminated the sense of flicker. So here's a point at which the 660 nanometer light was adjusted to match the 500 nanometer standard and completely eliminate the sense of flicker, and therefore gave us the ratio of sensitivity of that one pigment to those two wavelengths. And then we just do the same thing for the other wavelengths. So here we get, by this method, the absorbance spectrum of the M pigment in the living human eye. And out here, in yellow and red, are two polymorphic variants of the L pigment, the longer wave pigment. As I haven't mentioned this yet, but as I'll mention now, it does come in two flavors depending upon which amino acid, alanine or serine, occupies position 180, and that gives about a 4 nanometer shift. Again, a very common variation in our species. In fact, about 60% of human X chromosomes have the serine version, about 40% have the alanine. So in this room, for example, although many of us think we are, quote, color normal, actually we divide up perceptually into different groups depending which polymorphisms we have. Okay, now for the main course, the evolution of primate color vision. So first, how do we know that an animal has color vision? Obviously, with a person, they can tell you. You can ask them to discriminate various stimuli. With an animal, we do an analogous test, which of course is nonverbal, and what we do, and when I say we, I mean Jerry Jacobs, whose picture is shown here. This is Jerry. We don't usually show the animals the picture of the person, but rather a blank panel of a given hue. But the idea is illustrated here, and that is this little monkey is trying to decide which of the three stimuli looks different from the other two. We're not asking the philosopher's question, what does it look to you? We're just asking, can you discriminate? And that's the classic approach in color vision psychophysics. And if she pushes the button over here to the right, she'll get a little candy in this candy receptacle. So by this method, we can figure out what animals discriminate. And of course, if you do that, you will be able to map out the color vision abilities of different species. And what you find is that old world primates, like ourselves, like chimps, macaques, and so on, orangutans, have trichromatic color vision. New world primates, South American primates, don't all have color vision. Only some of them do. And let me just defer for a few minutes the discussion of who has and who does not have color vision among the New World primates. But we can ask, in either case, what's it good for? Why have the primates acquired color vision when all of the lower mammals, dogs, cats, mice, cows, pigs, and so on, have only dichromatic color vision? Well, the answer is we don't know. I mean, one never knows with these evolution questions, but there's some good guesses. One of them is that if you want to find ripe fruit among dappled foliage, that dappled in the sense that the light is sort of filtering through and the intensities are variable in kind of a heterogeneous, micro-heterogeneous way, being able to discriminate wavelengths, especially at the long wave end of the spectrum, where the difference between unripe and ripe fruit is very prominent, green versus yellow versus orange versus red, having trichromatic color vision is a huge help. And in fact, people who are dichromats have an especially difficult time picking out fruit like this from the unripe fruit. Of course, there are other possibilities. Looking at the appearance of one's fellow animals, especially if they're colored like this one is, could benefit from having trichromatic color vision. The answer is we really don't know, but it's clear that color vision of the sort we have arose at some point in the proto-primate lineage in the old world, and it's spread through and has been maintained in essentially all of those species, so it's presumably good for something. Okay, now let's just focus this question a little more, not on the evolution of trichromacy per se, but on this latest step, that is the acquisition by primates of a second long wavelength sensitive receptor. So here is that acquisition as seen from the sequence comparison angle. These are dendrograms showing the amino acid sequence identity of the different visual pigments, where the horizontal bar indicates the percent divergence. And here's the human case. The human L and M pigments are extremely similar in sequence, as shown by these short horizontal bars, and they're rather different from rhodopsin and from the S pigment. And here just on the vertical axis are the wavelengths of maximal absorption. And here is the mouse situation, which is not atypical for lower mammals. There's a single longer wavelength cone sensor. In this case, it's not that long wavelength. It's about 5-10 nanometers. There's a rhodopsin, and there's a shorter wave one. In this case, unusually short. It's actually down in the ultraviolet. But the basic arrangement holds for other mammals. That is, 2 cone pigments, 1 rod pigment, as compared to our 3 cone pigments. So now, the question is, how did this evolution occur? What were the steps, and how did it give us trichromatic color vision? And let's just consider for a second if we can break that down into various sub-parts, this evolution business. I think, obviously, one part is one has to evolve a new gene by gene duplication that encodes a pigment with a novel absorbance spectrum. That's pretty obvious. The second part is that one has to arrange for that pigment gene to be expressed in a subset of photoreceptor cells that are distinct from the subsets that are expressing the existing pigments. Because if they're all mixed together, there's no way to extract the different color information from a mixture of pigments in the same cell. And the third thing, which is maybe a subtler point, is one needs the neural machinery, the downstream wiring, to make the comparison of outputs between the different types of photoreceptor cells. It won't do just to have the new photoreceptor cells wire up in the same way as the old ones did and have their signals be mixed indiscriminately with the signals from the original cells. Okay, so we have these sort of 3 steps that have to happen, not necessarily in that order. And let's just take them one at a time. Okay, so we've got a gene duplication on the X chromosome. We've got a new gene. Now, in what way is it different to affect a different absorbance spectrum? Well, that we know something about. We know from the photochemistry of retinal and related compounds that the absorbance spectra of this class of compounds is very much determined, in fact, essentially exclusively determined, by the degree of pi electron delocalization. Now, these compounds, like all conjugated compounds, exist in a mixture of resonance forms. The most stable one is shown here, but you can certainly draw alternate forms. One is shown here. We've just shifted the electron over to the right, and we've got a free electron pair on the nitrogen. We've got a positive charge on this carbon. You don't have to remember very much college chemistry. Probably T and I, we got this part right in that recitation section. This ain't good, because carbons don't like to be positively charged, and this will be a minor resonance form. Now, just to show you the extent to which resonance plays a role, let me, and I'll just give away the punchline here. The more pi electron delocalization, that is, the more equivalent these 2 forms might be, or one could make them, the more redshifted the absorbance spectrum. And the less pi electron delocalization there is, that is, the less resonance there is, the more blue shifted the absorbance spectrum. And let me just show you some chemical data that supports that. If we look at two classes of model compounds, polyenes on the top and cyanins on the bottom, you can see this in its extreme form. So here's a polyene with the same number of double bonds as 11 cis-retinal, 6 double bonds. If we draw one of the resonance forms, an alternate resonance form as shown at the bottom, we can see this is extremely unlikely. We've put a positive charge on one end, a negative charge on the other, and we've shifted electrons over by 1 unit. This will make a minimal, if negligible, perhaps, contribution. And therefore, we can be confident that the polyene exists essentially in this upper form. This is true. How do we know it's true? Because if you look at crystal structures of polyenes, these double bonds are very short in the crystal structure. They are canonical double bond lengths. And these alternating bonds are very long. They're canonical single bond lengths. So here is a compound with an extreme alternation of bond lengths, hence minimal pi electron delocalization. If we look at cyanins, we get the polar opposite of that. I think you can appreciate that the resonance structure shown at the top here and the one shown at the bottom here must have identical energies, because the one at the bottom is simply the mirror image, left to right, of the one above it. So obviously they must be equivalent in energy. And in fact, there's maximal pi electron delocalization. And we know this because a crystal structure of a cyanin of this sort will show bonds of exactly the same length all down the chain, midway between single and double bonds. So these are compounds with maximal pi electron delocalization. Now this has a huge effect on the absorbance maximum. The polyene absorbs at 360 nanometers in the ultraviolet, and the cyanin at 750 nanometers in the far, far red. And you can think of the whole visual pigment spectral tuning problem as simply a question of making the 11 cis-retinal chromophore, which, if you have a close look at it, you realize one end looks like a cyanin and the other end looks like a polyene, making it look more polyene-like if you want a blue-shifted pigment, or making it more cyanin-like if you want a red-shifted pigment. And how does it work? Well, we pretty much know the answer. It works by placing dipoles, fixed dipoles, in the form of amino acid side chains around the chromophore. In the case of the human L and M pigment difference, there are 3, and only 3, that are major players. There's a phenylalanine tyrosine difference. These have a dipolar moment difference. An alanine-threonine difference, and an alanine-serine difference. In each case, a hydroxyl group has been added to the amino acid, and that adds a substantial dipole moment, and that causes a redshift. And so, in a sense, this aspect, this one of the 3 steps in the evolution of trichromatic color vision, I think is largely solved. We know the amino acid differences, and we know what they do to spectral tuning. A somewhat tougher nut to crack is the one about how you express a given visual pigment gene, a new visual pigment gene, in a distinct subtype of photoreceptor cells. Now, of course, there are many ways to do this in biology. The classic way is to evolve a promoter sequence that is distinctive, transcription factors that bind that promoter, et cetera, et cetera. But that seems not to be the case for the L and M pigment genes. First, their promoters are nearly identical. And second, there's a happy accident that the genes are on the X chromosome, and that seems to be the dominant fact that has controlled the mechanism being used here. So let me take a very slight divergence from the main topic of human color vision to talk about New World primate color vision, the color vision of South American monkeys. And they do this gene expression business by a mechanism that is entirely dependent on the X chromosomal localization of the pigment genes. So let me just remind you that among mammals, X chromosome inactivation in females controls the expression of X, or not all, but most X-linked genes, such that one of the 2 X's is inactive in any given somatic cell, and the genes on that X, roughly 80 or 90% of them, are turned off. And here in this female cat, we see one of the coat color genes where this has happened, and she's sort of a mosaic, a patchwork of expression of one X versus the other in her superficial cells. Now, if we look, and this is the work of Jerry Jacobs and John Mullen, if we look at the New World primates and ask about their color vision, we see a very striking pattern. And this is actually Jerry's initial discovery from the 1970s. Trichromatic color vision is seen only in females among New World primates. Dichromatic color vision invariably is found in males and is also found in some of the females, about 1/3 of them. And a typical New World primate, like these capuchin monkeys, would have the following arrangement. They would have a single X-linked visual pigment gene. They also have a shorter wavelength pigment gene that's on an autosome, as ours is. But that gene comes in 3 different alleles in the gene pool. And those alleles are present at roughly 1/3, 1/3, and 1/3 of the population. And they absorb maximally at either about 535, about where our middle wave sensor absorbs, about 550, roughly midway between the middle and long wave sensor, and about 563 near our long wave sensing pigment. And the females have got their trichromatic color vision because they are the ones that are heterozygotes for 2 different polymorphic variants. And because of X inactivation, their retinas are mosaics. And so within a single retina, they are expressing 2 different kinds of longer wavelength sensors thanks to this mechanism, and therefore can discriminate along with their shortwave pigment gene as a trichromat can. The males, of course, having only one of the 3 types, is a dichromat, but it's a dichromat of one or another sort, depending which one it has. Now, here's an interesting twist to the story. If we ask, by sequencing, again, this is Jerry Jacobs and John Mullen's work, and Jay Knight's and Maureen Knight's, by sequencing the DNA of these New World primates, we can ask, what is different in the DNA sequence that distinguishes these different allelic variants of their longer wave pigment gene? And the answer is, it's exactly the same amino acids that distinguish our long and medium wave genes. That is, positions 180, 277, and 285 can have either alanine or serine, phenylalanine or tyrosine, or alanine or threonine. And this is what argues for the allelic differences predating the duplication of our L and M pigment genes. These variants, we think, are the primordial primate variation that existed in all primates on the planet before Africa and South America moved apart by plate tectonic movements, and that were present in the proto-primate prior to that acquisition of the duplicated L and M pigment genes that we and other African-derived primates now have. And so let me just show you in this very schematic timeline. Here's time going from left to right. Way back when, there were lower mammals that had just a short and longer wave pigment gene, and those now have evolved to make all of the non-primate mammals that are on the planet. But that going down the primate lineage, there was initially a polymorphic variation that appeared in the X-linked photopigment genes, and that gave all of those animals, or at least the progenitors, the kind of color vision that New World primates now have. It's sort of the poor man's version of trichromacy, or perhaps I should say the poor woman's version, since it was only the women who had it. And then, subsequent to the divergence of New and Old World primates, that unequal recombination event occurred that gave us the duplicated X-linked pigment genes. So this is a sort of unusual example. When we think typically of genome evolution, we think of gene duplication followed by divergence. You make a second copy of your favorite gene, some random gene in the genome, and then since there's an extra copy, that is free to diverge in its sequence while maintaining the original copy in its own original job. This is the reverse. This is duplication following divergence. So divergence came first, duplication came second. Okay, now we're up to the last of the 3 questions. I think we have, or say we're up to the last of the 3 questions in the Old World primates. So we have now one mechanism to give cell type, no, I'm sorry, we're in the middle of the second question in the Old World primates. We have a mechanism that can give cell type specific gene expression in New World primates based on X inactivation. Curiously, it's a random process because X inactivation is random. So it gives a random sprinkling of different cone types in the retina. But now we have to ask, how about for us Old World primates who have duplicated these genes? Just having one of the 2 X chromosomes active doesn't solve the problem completely. Actually, it solves it partly, but not completely, of putting the product of one gene in one photoreceptor cell. How do we assure, if we have an arrangement like this, that only one gene will be active in a given photoreceptor type? Well, the X chromosome location has actually half solved the problem, because what it means is that each cell that has to make this decision only has to make it for one chromosome. You don't, if you're a female and have two X chromosomes, have to make the decision for both chromosomes since only one is active. So now we're down to, for both males and females, the question of how do you choose among the elements of a single array on a single chromosome to activate just one and not the others. And the first clue as to how this happened came from our study of individuals with blue cone monochromacy, a rare X-linked disorder in which both the L and M pigments appear to be inactive. And about half of families that have this trait have deletions in sequences that are upstream and adjacent to the L and M pigment gene array. And it turns out they're missing an essential enhancer element. We're going to call it a locus control region. And we know that enhancer element is critical. If we make a transgene that has a LAC-Z reporter downstream of it, we put it into mice, we get expression only in cone photoreceptor cells. If we cut out that little region of DNA, there's no expression at all. So it presumably cooperates with the adjacent promoter. And our model, and this is now a model, I think this is not at all proven, but just give me the benefit of the doubt here, let me just take you through it, is that this locus control region evolved a long time ago. I should say the sequence is highly conserved in all mammals as far as we can tell. It's been around for a long time. And it probably acted on the immediately adjacent promoter as any typical enhancer would. And this is especially appropriate in the Corey lecture, since he discovered enhancers, and here it is acting on a nearby promoter. And so in the ancestral mammals, there's nothing fancy going on here. This is just typical gene expression as we know it everywhere else in the body. But now with the presence of two nearly identical adjacent genes, this enhancer, which has not been duplicated, there's only one copy of it per human chromosome, per human X chromosome, the enhancer must choose between either the L pigment promoter or the M pigment promoter. And here, again, this is a model, we imagine that that choice is binary in the sense that it has to choose one or the other promoter, but not both. And also, we can further imagine that once the choice is made, it's stable. So this is the sort of thing that would give us an either-or kind of toggle switch for expression. Now, just to take this a step further, we could imagine various ways that this could happen. The classical way, the typical way that one thinks about is that, well, there might be transcription factors that originally evolved to mediate this sort of pairing, as shown by the black ball, and that these were around in ancient mammals and are probably still around today in present-day mammals that are not primates, and that in primates we have evolved additional transcriptional factor specificity such that one factor coordinates this pairing for the L cones, and a different factor, the green ball, coordinates it for the M cones, and then the transcription factors determine which pairing will occur. Okay, this is certainly possible, although I think from the point of view of explanatory power, it's just pushed the chicken and the egg problem back one chicken, because now we have to figure out what controls the transcription factors. But let me just present a completely different way that this could happen. One possibility is that the transcription factors have not changed at all in mammalian evolution. There is an ancestral transcription factor that generically mediates this sort of pairing between enhancer and promoter. The same factor exists in us, and the pairing event is stochastic. It happens on either the first or the second promoter, just as a coin toss. And once that pairing event has been set up, it's stable, and that particular gene will then be active in that cell in perpetuity. So I'll call this the stochastic model. We like this idea because it's a minimalist idea. That is, it doesn't require the evolution of some sort of complex apparatus, and there hasn't been that much time to evolve a complex apparatus. And it also accords with the random decision-making that the New World primates do. Obviously, exon activation is a random process. This hypothesis suggests another random process in New World primates. So how do you test this idea? Well, here's one test of it. If this idea is correct, then if we put the human L and M pigment gene array into a mammal that has never had to do this toggle switch decision, and we're going to use the mouse, the prediction is the mouse can do it. It's got all the apparatus necessary to choose either L or M pigment genes in its cone photoreceptor cells. So we did that experiment. This is Phil Smallwood and Yan-Shu Wang who did this. And so here's a human visual pigment array. It was put in at exactly 1 copy per genome. It's been fussed with a little bit. We put, instead of the visual pigment gene, the L pigment gene here, we put an alkaline phosphatase histochemical reporter, and instead of the M pigment gene, we put a LAC-Z, a beta-gal histochemical reporter. And then we ask, in the mouse retina, what's the pattern of expression of the alkaline phosphatase and the beta-galactosidase? Are they all expressed, are they both expressed as a mixture in all the cone photoreceptors or not? And the answer is very definitely not. Almost every cone that either expresses beta-gal in blue or alkaline phosphatase in brown, but not both. There's the occasional cell that makes both. And maybe that cell is in the act of switching, or there's some piece of this story that we don't understand. But the fact is, the mouse, which has never had to make this decision, is extremely good at making a mutually exclusive decision between L and M pigment genes if confronted with that pair. So that suggests that maybe it is stochastic. It's completely random. There is no designation coordinated with any other molecule in the cell except the visual pigment gene itself, whether this will be an L cone or an M cone. And if that's true, then of course that begs the question, how does the downstream cells, how does the wiring that is going to be connecting to these photoreceptors know the difference between an L and an M pigment containing cone? And I would suggest that, in fact, it doesn't know the difference. It just figures it out from experience. And the wiring, such as we know it, I think supports this hypothesis. If we look at the basic wiring diagram of a mammalian retina with respect to cone inputs, we see that in the ancestral arrangement, there is a shortwave cone limb in this circuit, which is opposed to the single pigment, I'll just call it M/L, the prototype of our M and L pigments, which is in the other limb. This is just a seesaw that takes one input as an inhibitory input, the other as an excitatory input, and essentially is taking the difference between them. These cells are well known, and they're present in essentially all mammalian retinas. The longer wave cones, in addition, at least in primates, where they're quite abundant, relatively speaking, also are used, and they're used primarily, for spatial vision. That is, there's a circuit which is comparing the longer wave cone type in one place in the retina to the longer wave cones immediately adjacent, and is using that for spatial discrimination. Now, as far as we can tell, essentially that same circuit has simply been co-opted for color vision information as well. So for example, what was the spatial circuit now pits M cones against adjacent cones, which in many cases are L cones. Actually, it's probably a mixture of M and L cones on this limb, and L cones against a mixture of M and L cones on this limb. As far as we can tell, the wiring is completely haphazard, but of course, because some cells will take as their input more M cones as excitatory and more L as inhibitory, or vice versa, and in the central fovea, where one limb takes only a single cone as single M or L as, let's say, the excitatory input, and the surrounding cones, a mixture of M and L as the inhibitory input, there will always be, in this sort of circuitry, some sort of chromatic differencing, as well as a spatial differencing. And very likely, those signals, the chromatic signal and the spatial signal, are being sent down the same wires and simply have to be sorted out in the cortex. Now that sort of neatly sidesteps the third problem, and it suggests, if this is all true, that there's no dedicated circuit that's needed for chromatic discrimination between L and M pigments. And as we just suggested earlier, there's also no dedicated transcriptional machinery that's required for the L versus M gene expression choice. This is now stripping the problem down to what seems to be a very simple set of answers. And if all of that is true, then it should be possible, and this is something that Jerry Jacobs and I started thinking about a decade ago, it should be possible to engineer that first step in the evolution of trichromacy into a mammal that has never had trichromatic color vision simply by adding an extra visual pigment gene. And all the other steps will take care of themselves. So we did that experiment. We put into the mouse genome, in place of the M pigment gene on the X chromosome, the human L pigment gene. So now when we look at heterozygous female mice who have on one X the normal M pigment gene, absorbs about 510 nanometers, and on the other X, the human L, which absorbs about 560 nanometers, we can ask, are they now endowed with a new dimension of chromatic discrimination? And the test, of course, is a behavioral one, the classic forced choice test that I showed you earlier, in which this mouse is deciding that that panel looks different from these two, and she's getting a little reward. It's a drop of soy milk, which they love. And if we do this sort of testing, and I should say a critical control is we randomize the intensities. So the animal cannot cue on intensity. It can only cue on chromatic differences. So as the intensities vary at random, it has to figure out, based on the chromaticity, what the difference is. If we look at the mean percent correct in this sort of forced choice test, where of course 1 out of 3 is correct. So if you know nothing, you can guess and get it right 1 out of 3 times, and that's shown down here. These are the mice that are the original wild-type mice or have only the human L pigment mice. They quickly realize that guessing is better than sitting back and doing nothing. So they guess every time, they get it right a third of the time. But if they have both L and M pigments, they get it right about 75% of the time. And this is now done, in this particular example, with a test wavelength at 600 nanometers that was pitted against a variable wavelength that was, as indicated here, every 10 nanometers from 500 to 600. As long as the two wavelengths are substantially different, they'll get it right about 75% of the time. If the two wavelengths are very close, say 590 nanometers versus 600 or 595 versus 600, they can't discriminate. But what this says is that simply adding a new receptor gene at the front end and allowing its expression via exon activation to be distinct spatially from that of the parental pigment, so that is we have a mosaic of L and M cones in the retina. That's all you need. The mouse is quite capable of harvesting that information and making useful discriminations based on it. So I think it argues that the name of the game here is plasticity. That is, the brain is endowed with an innate plasticity that can use new information at the front end of a system. And I think it also would argue that in the real evolutionary event, whenever that happened 50 million years ago, that primate that first got a variation in its X-linked visual pigment gene, the female primate that was a heterozygote for that variation, immediately saw a world of color that no primate had ever seen before. Thank you. I'll stop there and take any questions.
Over here. So my first question is regarding the allelic variants of L and M pigments in human. So I'm thinking about the evolutionary significance of these allelic variants. So I'm trying to imagine that human during the course of their evolution have started to use different hues of colors as compared to other old world monkeys. So this being able to see those would be useful for them. So then my question is, do old world monkeys also have different variants of L and M cone or not?
They're pretty consistent across old world monkeys. So macaques have virtually the same set of visual pigment absorbances that we have. I mean, I think our environment maybe in the last 10,000 years has been different, but up until that time, maybe it wasn't all that different from what it was in the preceding few million years.
And the second question is regarding the LCR. How do you imagine that the LCR, which is stochastically choosing one or the other gene, stays stable for the rest of the life of the cone with that gene?
Yeah, that's a great question. We really don't have the answer. Unfortunately, cones are rare in the primate retina, relatively speaking. They're intermingled with other cell types, and it's not an easy system to do biochemistry, for example. But I think you can imagine, for example, that an initial pairing interaction would, for example, allow the other promoter to become methylated. You know, some indelible mark that would then make the pairing event essentially permanent. But we really don't have any data on that.
Thank you.
Anand.
So, Jeremy, if the choice of L versus M is stochastic, then in that case you must have almost 50% of L and 50% M cones. I mean, the ratio should be almost equal in general, but that is not the case. There is tremendous variation that would suggest that human genome does have variations in some sort of other factors which indirectly or sort of in some way control the choice between L and M in addition to what LCR is doing. I mean, that's one way of looking at it. And the second question, which is related, is that in all cones, I mean, there's only one visual pigment is expressed, and the third one, which is complicating here, is the blue cone pigment, is the S-opsin. So somehow the cell also knows that it not only has to sort of turn on L or M, but also turn off S. So how are you imagining, how are you sort of trying to think through these problems, and what are your views? What do you think is happening?
I think, let's take the second one first. I think the LM versus S difference, that's a very ancient difference, because those pigments, those separate families have been around for a long time. The promoters are very different. The photoreceptors themselves have differences other than pigment content. For instance, their spatial arrangement, their abundance, and so on. So I think that is probably solved in sort of the same way that genes in general are differentially expressed. There'll be specific transcription factors and so on. I think the L versus M, or the L to M ratio difference that you described, so just to bring everyone up to speed here, that ratio is variable from person to person over quite a large range. Say, I think the most extreme range is about 10-fold, but the typical range is maybe 2 to 3-fold, still quite substantial. So, I mean, no one knows why that is. One possibility is that there are sequence differences that are in cis that affect that variation, although no one knows what they are if that's true. But I think the choice of L versus M is clearly very dependent on local factors. For example, the next gene in the array, which is an M pigment gene in most people, which looks just like the first M pigment gene, is never picked, or virtually never picked. So a distance, so this is a pairing mechanism that can go about 5 kb, it can go about 45 kb, but for some reason it can't go 85 kb. So you could imagine all kinds of chromatin effects that would affect the distance or geometry abilities of that pairing mechanism that could change the ratio, but it's totally unknown.
Yeah, but the first answer that you were saying, that LM versus S, is probably not as simple transcriptional activation, because if you turn on L or M in the cone, you also have simultaneously some mechanism to turn off S, and vice versa. So whatever is turning on, for example, S, has to turn off L or M, right? Because so these things are not independent. Somehow one will have to imagine some cross-regulation.
But it could be at a very high level. I mean, for example, L and M cones they're probably tagged synaptically in a way that's different from S cones. So there's probably some, higher order transcriptional control which feeds down onto many genes, of which the visual pigments are just a subset. That'd be my guess. But you're going to solve this, not me.
Okay, so in the interest of time, those of you with questions for Jeremy, we do have a reception for all of you to meet with the speakers. So maybe you can ask him right in front of the lobby by the library. Okay, so thanks again, Jeremy, for a wonderful lecture, and we really enjoyed your coming.